Without a way of terminating the pharmacologic action of drugs, the effects of drugs will persist indefinitely, and this, of course, will not be acceptable. Two factors are involved in terminating the pharmacologic and biologic effects of drugs, and these two factors are renal excretion and metabolism. By far, the most important quantitatively is metabolism. And why is metabolism more important than renal excretion? To start with, most drugs are lipophilic, and the lipophilic nature of the drug ensures that after they are filtered at the glomerulus, most of the drug will be reabsorbed at the nephrons, so they are still retained in the body. Another thing is that a lot of the drugs are tightly bound to plasma proteins, and because they are tightly bound to plasma proteins, they will not be filtered at the glomerulus. So the more important of the two is metabolism, and that metabolism will be the topic of our discussion in this presentation. What is metabolism? Metabolism is a series of processes that convert drugs into water-soluble and easily excretable products in the body. It is the biochemical changes that the drugs and other foreign substances in the body undergo. And metabolism, other words that I use interchangeably with metabolism, biotransformation or detoxification. And metabolism leads to the formation of different metabolites with different effects. Most of the time, metabolism will lead to the generation of pharmacologically inactive metabolites. However, there are cases when metabolism will lead to generation of metabolites that are active or even more active than the parent drug. And also, there are drugs that are called prodrugs, which are administered in the inactive form, but through the process of metabolism, those drugs will now be converted to the active products in the body. The majority of metabolism takes place in the liver, but some can occur in other tissues. For example, the gastrointestinal tract is an important place for metabolism of a lot of drugs. And when we talk about first-pass effects, we'll discuss more about metabolism of drugs by the GIT. Then other organs in the body also contain metabolizing enzymes and are important in metabolism of some drugs. For example, the lungs contain metabolizing enzymes and some metabolism take place in the lung. Same thing with the kidneys, with the skin, and still in some other structures in the body. But the majority of metabolism take place in the liver. The reactions that take place in metabolism are categorized into two. And these two categories are phase 1 reactions and phase 2 reaction. Now when you hear phase 1, then phase 2, it will appear as if they follow each other chronologically. Yes, in most cases, that is what happens. However, in some instances, the phase 2 reaction will actually precede the phase 1 reaction. And an example that really comes to mind in this is the case of the antituberculous drug isoniazid. Isoniazid first undergo N-acetylation, which is a phase 2 reaction, and then the product of that phase 2 reaction will now be subjected to a phase 1 reaction. There are other drugs like that, and we mention some of them as we go along. Why is metabolism important? It is important because it terminates the biological and pharmacological activities of drugs. An example of inactive or prodrugs that are converted into active compounds in the body by metabolism are enalapril, which is converted to the active compound enalaprilat. Another example is levodopa, and yet another example is benorilat, which is converted to aspirin and paracetamol by the process of metabolism. Phase 1 reactions are otherwise referred to as functionalization reaction. And basically, what happens in phase 1 reactions is that there is unmasking of a functional group or addition of a functional group on or to the parent drug. And these functional groups include OH, NH2, and SH. So the addition of these functional groups to the parent drug will lead to the formation of polar compounds that are easily excreted by the kidneys. However, in a lot of cases, the products that are formed are not polar enough to be excreted freely by the kidneys, and they have to undergo a phase 2 reaction. Phase 1 reactions generally result in loss of pharmacologic activity, and the most important enzyme involved in phase 1 reaction is the cytochrome P450 enzymes. Examples of reactions in phase 1 are oxidation, reduction, hydrolysis, cyclization, and decyclization. Phase 2 reaction, on the other hand, are referred to as conjugation or synthetic reaction. And in phase 2 reaction, what happens is that there is a formation of a covalent bond linkage between a functional group on the parent compound or a phase 1 metabolite and endogenous substrates like glucuronic acid, sulfate, glutathione, amino acids, or acetates. It leads to the generation of polar functional groups that can be eliminated in the urine, and this is catalyzed mainly by transferases. And examples of phase 2 reactions are glucuronide conjugation, acetylation, methylation, sulfate conjugation, glycine conjugation, glutathione conjugation, ribonucleoside nucleotide synthesis. N-acetylation is important in the metabolism of drugs like isoniazid, hydralazine, caffeine, dapsone, nitrazepam, and others. And two acetylator phenotypes are described in humans: the fast acetylators and the slow acetylators. So simply, drugs that are metabolized by acetylation are metabolized faster in the fast acetylators, while the metabolism is slower in slow acetylators. An important example is the antituberculous drug isoniazid. Glucuronidation. What happens here is that glucuronic acid is transferred from UDP glucuronic acid to a substrate, and it is important in the metabolism of some benzodiazepines of chloramphenicol, paracetamol, bilirubin, thyroid hormones, and steroid hormones. And a lot of drugs excreted, a lot of drugs that undergo glucuronidation are excreted through the bile, and from there they undergo enterohepatic recirculation. Glutathione conjugation is important in paracetamol metabolism. Sulfation is carried out by enzymes referred to as sulfotransferases, of which 11 isoforms have been identified in human beings, and it often leads to generation of chemically reactive metabolites. It should also be noted that significant fractions of circulating catecholamines, estrogens, iodothyronines, dihydroepiandrosterone exists in the sulfated form. Ribonucleoside nucleotide synthesis is important in the activation of many purine and pyrimidine antimetabolites that are used in cancer chemotherapy. What are the factors that influence metabolism of drugs? Chemical structure of the drug is important. Species differences is important. Physiological or disease state is important. Genetic variations is also important. Then age is also important. Age is important because the activity and the concentration or the amount of metabolizing enzymes are not fully developed at birth, and they also decline in the elderly. Also, the amount of the endogenous substrates that are important in metabolism might be less in both the young and also in the elderly. Sex is also important, and it's been noticed that drugs like diazepam, caffeine, and paracetamol are metabolized faster in females. Enzyme induction or inhibition is also important. Then competition for endogenous substrates is also important. Diseases like acute or chronic hepatic parenchyma diseases can reduce the concentration of metabolizing enzymes, since these enzymes are synthesized by the liver. Cardiac diseases also can inhibit metabolism by reducing hepatic blood flow. Pulmonary disease is important in the hydrolysis of procainamide and procaine, and their hydrolysis is impaired in chronic respiratory insufficiency. Thyroid dysfunction also, both hyperthyroidism and hypothyroidism have important effects on the metabolism of certain drugs. Genetic factors is also important because it can influence enzyme levels or lead to the formation of abnormal forms of enzymes. Examples are the genetically determined defects in pseudocholinesterase, which affects metabolism of succinylcholine, then acetylation of isoniazide, hydroxylation of warfarin, and others. The drugs that are responsible for the metabolism, the enzymes that are responsible for the metabolism of drugs are classified as microsomal and non-microsomal enzymes. The microsomal enzymes are otherwise known as the microsomal mixed function oxidase enzymes, or simply monoxygenases. These are found in smooth microsomes, and they are responsible for oxidative drug metabolism. There are two main enzymes in this class, and these are NADPH cytochrome P450 reductase, which is a flavoprotein, and cytochrome P450, otherwise referred to as CYP or P450, which is a hemoprotein. The P450 is quantitatively more important than the cytochrome P450 reductase because the P450 is responsible for the metabolism of most of the drugs that are taken by human beings. There are several isoforms of the P450, but again, the quantitatively most important one among them is the CYP3A4, which is responsible for metabolizing over 50% of the drugs that are metabolized by the liver. It's important for us to know that these enzymes can be induced or inhibited. And what induction simply means is that there is enhancement of the activity of the particular enzyme under discussion. Induction can be by enhancement of the rate of synthesis of the enzyme or by reduction of the rate of degradation of the enzyme. And induction leads to more rapid metabolism of substrate and any co-administered drug. And substances that can induce the microsomal enzymes include barbiturates, phenytoin, carbamazepine, rifampin, and others. Now, what are the effects of enzyme induction? Number one, it leads to decreased intensity and or duration of action of drugs that are inactivated by metabolism. It also leads to increased intensity of action of drugs that are activated by metabolism. It can also lead to the development of tolerance. Then, importantly also, some endogenous substrates such as steroids and bilirubin are metabolized faster due to enzyme induction. These are some of the other possible effects of enzyme induction. Is this enzyme induction all bad? No, it's not all bad. It has its uses also. In certain situations, it can be beneficial. For example, in congenital non-hemolytic jaundice, enzyme induction is beneficial. Then in Cushing's syndrome, in chronic poisoning, and also in liver disease, enzyme induction are beneficial. Enzyme inhibition, here, the activity of the enzymes are attenuated. And this can be by competitive inhibition or by the enzyme being rendered catalytically inactive. And drugs that can inhibit the enzymes include cimetidine, ketoconazole, erythromycin, itraconazole, ritonavir, and others. For the non-microsomal enzymes, these ones are found in the cytoplasm and also the mitochondria of hepatic cells, as well as in other tissues, including plasma. And examples are esterases, amidases, and others. And reactions that are catalyzed by them include some oxidation and reductions, many hydrolytic reactions, and all conjugations except glucuronidation. Both the microsomal and non-microsomal enzymes are deficient in the newborn, especially the premature, making them more susceptible to many drugs. Apart from metabolism leading to the generation of drugs, of metabolites that are inactive, or from converting inactive drugs to active drugs, it can also lead to the generation of toxic products. And this usually happens when the main pathway for the metabolism of the drug has been exhausted. And when this happens, the metabolism will now shift to alternative pathways, which are usually toxic. A good example here is paracetamol. Paracetamol at standard doses, when it's taken at standard doses, the liver is able to cope with its metabolism. But when overdoses or overdosage occur, then the endogenous substrate, which is responsible for the metabolism of paracetamol, will eventually get exhausted. and the alternative pathway will now take over. And this alternative pathway for paracetamol metabolism is the P450 dependent GSH conjugation pathway. So with depletion of GSH, there will be generation of toxic metabolite. And the toxic metabolite is N-acetylbenzoiminoquinone. So this toxic metabolite will react with hepatic cellular protein nucleophilic groups to cause hepatotoxicity. So in cases of paracetamol poisoning, the antidotes to use are cysteamine and N-acetylcysteine. Thank you for listening.